So now let us see about another group of compound lipids, which is glycolipids. The name itself suggests glyco means carbohydrate and lipids are lipid molecules. So you have carbohydrates combined with lipid molecules. And these compounds are a group of compound lipids. Let us see the various composition of this particular glycolipid. You have fatty acid which is attached and a sphingosine which is a 19 carbon amino alcohol, and you have a carbohydrate. And remember what we said, fatty acid plus sphingosine? Yes, you are right. It is ceramide. Fatty acid attached to a sphingosine is ceramide. Along with that, if you have carbohydrates, it is a glycolipid. And because most or nearly all the glycolipids have sphingosine as an alcohol, they are referred to as glycosphingolipids. And the major function of this glycosphingolipids is they are a component of cell membrane as all lipids, and also they are important constituents of the nervous tissue, and they are used or important for the development of nervous system. There are different types of glycolipids. We have something known as cerebroside. The other one is globoside, and you have ganglioside. So what are these cerebrosides, globosides and gangliosides? They are various groups of glycosphingolipids or glycolipids. Cerebroside, the basic structure of fatty acid and sphingosine, that is a ceramide structure, is common for all the three groups. Okay, so you have ceramide in all the three types of glycolipids. In addition, if you have a monosaccharide, then this is referred to as cerebroside. The monosaccharide units may be glucose or galactose. So glucose cerebroside or galactose cerebroside, they are important for the development of nervous system. And you have globoside, where ceramide is attached to more than one monosaccharide units. So here if you see, you have a ceramide and an oligosaccharide. So the most common example is lactosyl ceramide. You know what is lactose, right? It is a milk sugar which is made up of galactose and glucose. So this glucose and galactose is attached to the ceramide. It is referred to as lactosyl ceramide, and it is present in the RBC membrane. Then you have something known as ganglioside. What is special about this ganglioside is, in addition to the ceramide and the oligosaccharide or the sugar moiety, you have a substance which is N-acetylneuramic acid. N-acetylneuramic acid or NANA. This is also referred to as sialic acid. N-acetylneuramic acid or sialic acid. This is the ganglioside. Now, why do we read all these glycolipids? There are much clinical relevance with reference to our nervous system. In addition, there is one particular clinical relevance with reference to a bacterial infection, that is cholera. You all would have known the typical rice water stools present in cholera, which is a bacterial infection. So how does that rice water stool or watery diarrhea occurs in cholera? Is because of the action of this GM1 ganglioside. So we have the cholera toxin. The cholera is a bacterial disease caused by Vibrio cholerae, and this produces a cholera toxin which is made up of two different subunits, A subunit and the B subunit. The B subunit is going and binding to the ganglioside receptor which is present in the intestinal epithelial cell, and this is most commonly the GM1 ganglioside. So now, and in addition, the A subunit will go and activate your G protein. G protein will activate your adenylate cyclase, and adenylate cyclase will lead to the synthesis of cyclic AMP. Cyclic AMP is going to go and activate many of the protein kinases. So kinases are enzymes involved in phosphorylation. So this phosphorylates many of the channels and ions required for all the transport of various ions. And one such is the chloride channel ion. So this leads to phosphorylation of the chloride channel, and that leads to efflux of the chloride from the epithelial cell into the intestinal lumen. In addition, sodium and water are also pulled into the intestinal lumen. So as a result, all the electrolytes are poured into the intestinal lumen, and this leads to the watery diarrhea associated with electrolyte imbalance in case of Vibrio cholera. Let us see a few important short answers related to few of the properties of lipids. One such thing, we all know lipids are fat molecules. So we have something known as oils and fats. What is the difference between an oil and a fat? Oils are nothing but they are liquids at room temperature, whereas fat is a solid at room temperature. And oils have more amount of unsaturated fatty acids. You remember unsaturated fatty acids? They have the presence of a double bond. And here, in case of fats, you have more saturated long-chain fatty acids. And oils are mostly of plant origin. You have sunflower oil, corn oil, olive oil, etc. And fats are more of animal origin. You have butter, ghee, etc. So this is one important classification which you need to know for a short answer. Then let us see an important property of lipid, which is saponification. Triglycerides, we have seen the glycerol molecule attached to three different fatty acids or three same fatty acids. Triglycerides or triacyl glycerols are hydrolyzed by alkali to form glycerol and soap. This process is known as saponification. And we have something known as saponification number, which is a very commonly asked short note, a 2-mark question, or it can be asked in an MCQ. This is a number of milligrams of potassium hydroxide or the alkali which is required to saponify 1 gram of fat. That is saponification number. Number of milligrams of the alkali or potassium hydroxide required to saponify 1 gram of fat. So why do we need to know about the saponification number? It is an indicator of the molecular weight of fat and it is inversely related to the molecular weight. So if you have a higher saponification number, the molecular weight is less. If you have a lower saponification number, the molecular weight is high. Let us see the saponification numbers for few of the common lipids. Butter has a saponification number of 210 to 230 and coconut oil has a saponification number of 250 to 260. So here if you see, coconut oil has a higher saponification number and butter has a lower saponification number when compared to coconut oil. So this indicates the molecular weight inversely. Then we have another concept which is iodine number. This is nothing but number of grams of iodine taken up by 100 grams of fat. Number of grams of iodine taken up by 100 grams of fat. What does this number signify? It is an index of the degree of unsaturation. Unsaturation is the presence of double bonds. Higher the iodine number, higher the unsaturation. So higher numbers you get for this particular iodine number, you are going to assume that that particular lipid has more number of unsaturated fatty acids. Let us see a few examples again. Iodine number of butter is 28 and whereas that of sunflower oil is 130. So here sunflower oil when compared to butter has a higher degree of unsaturated fatty acids. You remember we have seen already the difference between oils and fats. Oils have more amount of unsaturated fatty acids. Then there is an important property of lipids which is known as rancidity. Rancidity of fat is nothing but the unpleasant smell and taste which is there when it comes to the fats and oils or when they are stored for longer periods. There are two types of rancidity, which is hydrolytic rancidity and oxidative rancidity. Hydrolytic rancidity occurs because of the hydrolysis of triacylglycerol molecules which lead to an unpleasant smell or taste. And oxidative rancidity is because of the partial oxidation of the unsaturated fatty acids to yield compounds such as epoxides and free fatty acids which lead to the development of this unpleasant smell or taste. Now let us see two important structures which are formed with relation to lipids. One is micelle and liposomes. These two are structures which are formed because of an important property of lipids which is amphipathic. We have already seen the word amphipathic related to phospholipids, right? Amphipathic is where you have a hydrophobic and a hydrophilic group together. So let us see what is a micelle and what is a liposome. Micelle is nothing but phospholipids which are distributed in water to form molecular aggregates. You know the structure of a phospholipid. Remember the hydrophobic and the hydrophilic region, the hydrophobic tail region and the hydrophilic head region. When it is distributed in a water containing medium, it tends to aggregate among itself where you have the hydrophobic part within the aggregate and the hydrophilic part on the exterior. So these micelles help in solubilization of the lipids in aqueous media. And also it assists in the digestion and absorption of lipids where you have all the lipids to form a micelle-like structure. All the hydrophobic goes inside and the hydrophilic outside. And this then it further proceeds to emulsification where you cut it down into small lipid droplets. Then let us see about liposomes. So the same phospholipids will form the phospholipid bilayer, and when several phospholipid bilayers aggregate, they form, along with the other amphipathic lipids, they form liposomes. These are spherical vesicles of one or more phospholipid bilayers. They are prepared by sonication of a mixture of phospholipid and cholesterol. Phospholipid is also amphipathic, cholesterol is also amphipathic. And the most important application of this liposome is it is used as a drug delivery system. The most common drug which is given in the liposomal form is amphotericin B, which is used in the treatment of fungal infections. In addition, liposomes are also used for targeted gene therapy. Now let us see the most important steroid group of lipids, that is our cholesterol molecule. What do you know about cholesterol? Cholesterol is going to lead to the development of various cardiovascular diseases, and it leads, and you all know that high cholesterol is very bad for health, right? So what does the word cholesterol mean? Chole means bile, steros means solid, and OL stands for alcohol. So you have a OH group also which is present. And this cholesterol is an animal sterol. It is a 27 carbon steroid ring. You have 27 carbons arranged in cholesterol molecule. The ring which is present is referred to as cyclopentanoperhydrophenanthrene ring, CPPP ring. So this is a very important MCQ aspect. So the ring structure present in steroid is cyclopentanoperhydrophenanthrene ring. Now let us see the structure of cholesterol, which has 27 carbons, right? So you have the A ring, B ring, C ring and D ring. 4 rings are there. That is a cyclopentanoperhydrophenanthrene ring, right? And if you see in the third carbon, you have a OH group attached. So this is going to be the hydrophilic part of our cholesterol molecule. Then if you see, you have a double bond between 5 and 6. So in the third position, OH group, and between 5 and 6, you have a double bond. And total number of carbons is 27. So here is a side chain. These carbons form the side chain of cholesterol. So because there is a OH group attached to it, it is a amphipathic molecule, and when this OH group is going to go away, that is, if this OH group is going to be esterified with another fatty acid to form cholesterol ester, that becomes completely hydrophobic. Now, what are the different functions of cholesterol? They are components of cell membranes, and you know what? This is responsible for the maintaining the fluidity of membranes. Also, it has an insulating effect on nerve fibers. Cholesterol is also involved in lipoprotein metabolism. You know, cholesterol has to be carried through various lipoproteins, that is HDL and LDL, high density lipoprotein and low density lipoprotein. And there are certain special compounds which are derived from cholesterol, which is a very important short note answer, or it can be asked in the form of MCQs. You have bile salts and bile acids, which are required for the digestion of lipids. Then we have the various steroid hormones. Steroid hormones include your aldosterone, you have those which are produced from the adrenal cortex, and then in addition you have the estrogen and testosterone, which is also going to be produced from cholesterol. Then you have a vitamin which is going to be derived from cholesterol, and that is vitamin D. Now let us see the clinical significance of cholesterol. You all know that cholesterol, when it gets accumulated within the blood vessels, it can lead to obstruction of the blood flow. So atherosclerosis, deposition of LDL cholesterol within the intima layer of the blood vessel, is referred to as atherosclerosis. It leads to thickening of the vessel wall, and as a result, when it progresses further, it can lead to obstruction of the blood supply. Then diabetes mellitus. Diabetes mellitus is one of the most common cause of increased cholesterol levels, and this, in other words, it has dyslipidemias, which we will be seeing in a short time. Then all these reasons, that is atherosclerosis and the deposition of this cholesterol in the blood vessels, leads to the development of cardiovascular diseases. Now let us see a short glimpse of the various bile acids and salts which are derived from cholesterol and which is required for the digestion and absorption of lipids. So bile acids are synthesized from cholesterol, and there are two different bile acids, that is primary bile acids and secondary bile acids. Primary bile acids are synthesized in the liver and secondary bile acids are synthesized in the intestine. Primary bile acids are nothing but they are cholic acid and chenodeoxycholic acid. These are synthesized from cholesterol in the liver, cholic acid and chenodeoxycholic acid. Secondary bile acids are synthesized in the intestine. That is, they are deoxycholic acid and lithocholic acid. So we have bile acids which are primary and secondary. Primary from the liver, that is cholic acid and chenodeoxycholic acid, and secondary which is synthesized in the intestine, which is deoxycholic acid and lithocholic acid. Now let us see a short glimpse of the steps of bile acid synthesis. So from cholesterol, the enzyme 7-alpha hydroxylase. This is a rate limiting enzyme of bile acid synthesis. Rate limiting enzyme of bile acid synthesis is 7-alpha hydroxylase. You don't need to know the complete steps, but whereas remember that cholesterol by 7-alpha hydroxylase will be converted to 7-hydroxycholesterol, and through a further series of steps, this is not just one step, a series of steps, you form the primary bile acids cholic and chenodeoxycholic acid, and these primary bile acids get conjugated to glycine or amino acid and taurine. Glycine and taurine, they get conjugated to glycine and taurine to form glycocholic acid and taurocholic acid. When chenodeoxycholic acid conjugates with this glycine and taurine, we get glycochenodeoxy and taurochenodeoxycholic acid. So when these glycocholic and taurocholic, glycochenodeoxycholic and taurochenodeoxycholic get conjugated with sodium or potassium and they form salts, they are referred to as bile salts. So far we have been seeing about the bile acids. They get conjugated to glycine or taurine, and these, when they combine with as sodium and potassium salts, they form the bile salts. So after this particular bile acid gets secreted into the intestine, then there it is acted upon by bacteria to form the secondary bile acids, which are deoxycholic acid and lithocholic acid. These are the secondary bile acids formed in the intestine by the action of intestinal bacteria. So the rate limiting enzyme of bile acid synthesis from cholesterol is 7-alpha hydroxylase. Now let us see the various functions of bile salts. Most important function is helps in the digestion of lipids, that is emulsification. What is emulsification? The process of breaking down the large lipids into smaller droplets. Only then you can further enhance the absorption of lipids. So it helps in digestion of lipids. Then we have already seen the Michel formation, right? Michel formation is also a process of the digestion of lipids, where by the action of different enzymes, the molecules tend to form aggregates, where you have the hydrophilic exterior and the hydrophobic interior. So Michel formation. Then these bile acids also serves as regulatory molecules. That is, there is some receptor known as farnesoid X receptor, which helps in the regulation of gene expression.